Okay, hello everyone. Thank you very much for coming on the last day of finals week. So as Karin said, my research focused on ways to improve the outcomes of seed-based revegetation in an uncertain water future. So I'm going to cover some background information and the motivations for research, and then we'll look at both chapters of my thesis, which cover 2 greenhouse experiments that I did, as well as a field experiment in chapter 2. And then we'll talk about management recommendations based on my results. So the frequency and severity of extreme hydrologic events like drought or excessive precipitation are projected to increase over the next century worldwide due to climate change. And wetlands are especially vulnerable to these changes because hydrology is their defining feature. Wetlands only make up a small portion of the Earth's surface, but they provide really critical ecosystem functions and services that we rely on, like fire and flood mitigation. And native vegetation drives many of those functions and services. And changes in flooding regimes from drought or excessive precipitation is going to strongly influence plant community composition. So we've seen hydrologic extremes play out already in our study system of Great Salt Lake wetlands. That map on the left is from July 2022, when most of the state was in severe or extreme drought. And then after the record snowpack in 2023, that map on the right from July 2023 shows that it actually brought most of the state out of drought. So there's a lot of variability in water availability in these wetlands from year to year. And in addition to climate-driven changes from water availability, managers in this system are also contending with upstream water diversions from agricultural or municipal use that is limiting the amount of water coming into these wetlands. So native plant communities are essential to healthy functioning wetlands, and specifically in Great Salt Lake wetlands, they provide critical habitat and food for millions of migratory birds that pass through each year. There's a lot of species diversity in native Great Salt Lake vegetation, and there are a few broad plant community types that I'll talk about today that describe the hydrologic niches that all of these plants fill. So emergent species are plants that are rooting in standing water, and the vegetation is emerging out of the water. So they have adaptations to flooding and growing in anoxic environments. So meadow species grow in wet meadow wetlands. So the soil is saturated, but these are not inundated standing water conditions. So species in this group are adapted to a slightly drier environment. And then we also have playas around Great Salt Lake, which are seasonally flooded wetlands that dry down throughout the summer. So they leave behind really dry and highly saline soil conditions. So playa species have physiological adaptations to drought and high salinity that other species do not possess. So native plant communities are slow to return on their own in degraded wetlands, or they often will not return on their all. So that makes revegetation a critical part of the restoration process, but it is made more difficult by those hydrologic extremes that we're facing. Seed-based revegetation is where we spread seeds onto a landscape either by hand or through machinery, and it's scalable to really large areas, and it's often the most cost-effective and labor-efficient method of revegetation compared to hand planting seedlings or adult plants, which requires significantly more labor. But there's really high mortality in seeding events because seeds require specific conditions for germination, like adequate light and water. And after seeds do germinate, a lot of seedlings will not survive into maturity. And increasing hydrologic extremes are going to further threaten seedling establishment because they are much more vulnerable to extreme water conditions than mature plants. So because we're in a semi-arid system that is prone to drought, and we know droughts are projected to increase, we need to learn more about how these native plant species respond to drought and when they're grown from seed. And we need to know if any of them can tolerate or recover from drought. So this leads into the greenhouse section, where I wanted to find out generally how these native species are responding to drought, which species can best tolerate drought conditions, and if any species can recover from drought if water was returned at the end of a drought interval. So my first greenhouse experiment was just focusing on that first question of how does a community of native wetland plants respond to different lengths of drought. So the seed mix in this experiment consisted of these 5 graminoids in the top row, which are just grass-like plants, and then we had these 5 forbs in the bottom row, which are flowering broad-leaved plants. And we predicted that emergent species, which are the ones highlighted here, since they are adapted to flooding, we predicted these would be less tolerant of drought than the meadow species in the mix, which makes up the rest of the species here. So this experiment lasted 6 weeks, and we had 5 different hydrologic treatments. And these drought treatments were all aiming to mimic potential spring drawdown scenarios that we might see in the field. So for example, for 4-week drought, which is the top treatment here, this might be after a winter where we didn't have a lot of snow. Managers are going to be seeding in the spring when the soil conditions are still saturated, but we would expect after a dry winter, the site is going to dry out really quickly after seeding. So in this example, this treatment gives 2 weeks to these seeds to germinate before the drought sets in. And so for 3-week and 2-week drought, this is just a less severe version of that scenario. So we're mimicking a winter that had more snow. We would expect that site to remain saturated for a little bit longer, which gives those plants more time to establish before dry soil conditions set in. We also had a moist treatment, which served as our control. So the soil was just kept saturated through the experiment. And then we also had a flooding treatment just to serve as another comparison to the drought. So this was flooded with 5 centimeters of water on the soil surface for 3 weeks because that was the midpoint of the drought treatments. And 5 centimeters is very shallow, but since these were grown from seed, these are little tiny plants at that point. So oftentimes it's submerging the whole plant. So we mimicked drought by, we seeded it in containers that were filled with potting soil and then a layer of native wetland soil on top. And you can see those pictures on the right. And we watered these through a reservoir system. So those white planting containers were sitting in the reservoirs and water was wicking up the soil column. So to mimic drought, we just stopped adding water to the reservoirs and allowed the soil to dry out and did not add any more water for the continuation or for the entire duration of the treatment. So I collected visual percent cover data weekly for 6 weeks in 10% cover categories. And we also harvested dried and weighed biomass at the conclusion of the experiment. And I used mixed effect models to analyze the effect of hydrologic treatment on cover or biomass of both the total seed mix, each species individually at the end of the experiment. And we used a completely randomized design with 8 replicates. So we did not include random effects in these models. So these plots are showing you the raw data. This is the visual percent cover over time through all weeks of the experiment. The week of the experiment is on the x-axis, cover is on the y-axis, and then that gray shading in the background is indicating when a hydrologic treatment was taking place. That black line that the arrows are pointing to are showing us the total cover with all species combined. And in this experiment, cover started to decline 1 to 2 weeks after the onset of drought. This plot is the model estimated mean cover of these 4 species in the treatments. Cover is on the x-axis and treatment is on the y-axis, or backwards. Treatment is on the x-axis, cover is on the y-axis. The top left panel is showing us the total cover of the whole seed mix. So we only modeled these 4 species because we did not really see meaningful growth from the other 6 species in this seed mix. And we also did combine those 2 species in the bottom row, the inland saltgrass and the scratchgrass, into a perennial grass category because they were indistinguishable in the drought treatments at the conclusion of the experiment. And the general takeaway you can see here is that 4-week drought is often the lowest cover, and then it increases as the drought intervals shorten. And unsurprisingly, increasing drought lengths further reduce plant performance. So based on the results of this experiment, we still wanted to assess how these species, how resistant or resilient they are to drought. And the high performance of these 4 species influenced our methods for the second greenhouse experiment. So for this, we expanded our questions to also ask which species are the most resistant to drought, So which species can survive in drought conditions without declining in performance or with minimal declines in performance? Here is measured by cover or biomass. And we also want to know if any of these species can recover from drought if water is returned to the system. So we also split these species into a fast-growing and a slow-growing mix with different amounts of germination time to accommodate their different growth rates. We wanted to avoid those 4 species, which were the high performers in experiment 1. We wanted to avoid those completely suppressing the growth of the slower growing species, which is what I suspect happened in experiment 1, so that we can actually see the treatment effects on those slower growing perennials. So these are the same species in experiment 1, that the entire 10 species mix is included here, but we also included 2 playa species, which are those drought and salt tolerant species that I mentioned. And we predicted that drought tolerant species like the inland saltgrass and the pickleweed and perch seepweed, which are the playa species, would be the most resistant to drought, reflected through lower declines in cover, and they would also recover from drought the most effectively. So we fully replicated all of the treatments in experiment 1, but we added another set of treatments where after the end of the drought intervals, they were introduced to a 3-week recovery period where we returned moisture to all of the containers by top watering the soil with half a liter of water and then reflooding all of the reservoirs, because we wanted to see if any of those plants could bounce back from their drought stress state. So I again collected visual percent cover data weekly, but this time I split cover by both live and dead plant material so that we could get a better idea of how the plants are doing. So to determine or to categorize those, I looked at the bases of the stems on all of the plants. And if the stem was desiccated at the soil surface, that plant material was counted into dead cover, because we figured if it is that dried out, that close to the root, it's unlikely that it's going to be able to bounce back once water is returned. And that live plant material or live cover is what we analyzed in all of the cover models. But we also harvested dried and weighed biomass both before and after the recovery period so that we can compare biomass weights. So I used mixed effect models again to look at the effect of hydrologic treatment on cover or biomass of the total seed mix and each species individually, but this time we also included a random effective block with 5 replicates. And we also did a separate analysis looking at the effect of hydrologic treatment on and the week of the drought treatment on the difference in cover between the treatments and the moist control. So we wanted to quantify what we looked at with those cover over time graphs. So we want to know at which week of these drought treatments does cover begin to significantly decline. So we're going to go by each seed mix. These graphs are showing the live and dead total cover of the fast growing mix over all of the weeks of the experiment. And this is just total cover, so all the species combined. And the solid line is showing live plant material, which is what, again, is used in the models. Week of experiment is on the x-axis, cover is on the y-axis, and again, the gray background shading shows you when the hydrologic treatment was in place. So in experiment 2, in this seed mix, we saw that cover did not begin to decline until 2 to 3 weeks after the onset of drought. And in the 2-week drought, cover actually did not decline at all below its pre-drought levels. So these arrows are pointing to when the containers were in the recovery period. And in both 4-week and 3-week drought in this seed mix, as soon as water was returned to these containers, there was a ton of regrowth. This graph is the mixed effect model results for the resistance analysis. So the y-axis shows us the difference in cover between the treatments and the moist control. So above that dashed line at 0, more cover than the moist control at that point, and if it's below, it means it had less. And the x-axis is the week of the drought treatment, and so week 0 all the way to the left is the week before the drought treatments began to serve as our baseline. The purple points, which are outlined here, are the 4-week drought treatment, so the most severe, And the letters are telling us when there was a significant drop in cover. And in the fast growing mix, you can see we have a significant cover drop between weeks 2 and 3, and then again between weeks 3 and 4. So our drought treatments were just short term, but this shows us that even an extra week of dry conditions, once that resistance threshold is passed, will continue to negatively impact plant performance. So these are the model estimated mean cover graphs for the total seed mix and all of the species in the fast growing mix. The points in red are the data before the recovery period, and then the points in blue are at the end of the 3-week recovery period when everything had a chance to regrow. So the letters above the points are only showing significance within their color, so we're not comparing between the red and the blue, but only within the red and the blue. So this panel highlighted in red is the total seed mix, and though the 4-week drought estimate is still a bit lower than the rest of the treatments, there were few significant differences between treatments after the recovery period. So this is telling us that this seed mix overall was resilient to drought of up to 4 weeks and was able to return to the live cover estimates similar to all of the other treatments. So the golden dock, the species here, still had lower cover in after 4-week drought, after the recovery period. So this suggests its performance is negatively impacted by at least 4 weeks of dry soil conditions, but in 3-week drought, cover was still able to recover to a similar level as the moisture treatments. So 2 other species in this mix, the beggar's tick and the scratch grass, had no differences among treatments after the recovery period. So all of the cover levels were similar to the moisture treatments. And then one species, the inland saltgrass, had really interesting results. So this species actually had the highest live cover after it was exposed to 4-week drought. So in this case, it was not just resilient to drought. It seems that the drought actually stimulated its growth. And its second highest cover after the recovery period was in 3-week drought. These cover values are still low since it is a graminoid, but it did significantly better after 4-week drought than the other treatments. And we are going to go through the same information for the slow growing mix. So again, these are the plots of live and dead cover for the total seed mix. And the results here before the recovery period were pretty similar. Cover began to decline about 2 to 3 weeks after the onset of drought, but cover again did not decline in 2-week drought. So when the recovery period began, there was a small amount of regrowth from 4-week drought, but this seed mix did not recover nearly as well or as quickly as the fast growing mix. In 3-week drought, we had a little bit more regrowth during the recovery period, but it's still not quite reaching the peak of cover that we saw before performance began to decline. And then here is the graph for the resistance analysis again, showing the difference in cover. And the results here were really similar to the fast growing mix. And in 4-week drought, we saw cover drop from drought weeks 2 to 3, and then again from weeks 3 to 4. And then here are the model estimated mean cover graphs again. So with the fast growing mix, the recovery period, after the recovery period, there were few significant differences between treatments. But for the slow growing mix, we still had significantly lower cover in 4-week and 3-week drought compared to the moisture treatments after the recovery period. So this mix overall was not as resilient to the effects of dry soil and was not able to recover as effectively. So these 2 species had lower cover after the recovery period in both 4-week and 3-week drought than the other treatment or than the other species. Suggesting this could really only withstand 2 weeks of dry soil conditions before performance began to decline, and it could no longer regrow after drought. And this species on the right, pickleweed, and on the bottom picture here, is actually known to be a drought-tolerant species, so this result was really surprising. But this is also a species that tolerates and grows in really highly saline conditions. So I suspect here, since we used fresh water, that was impacting the performance of this species, and it might perform differently if it was in a higher saline environment. These 3 species performed similarly to the golden dock in the previous mix, with lower cover under 4-week drought, but cover was similar amongst all the other treatments after the recovery period. And then of all the species in the slow-growing mix, once again, cover is still very low since it is a graminoid, but with the species with the most significant cover here is the alkali bulrush, which had no significant differences between treatments after the recovery period. So just to do some takeaways, we predicted that the drought response would align with those plant community type classifications. So emergent species would not be able to tolerate drought as well as a playa or meadow species, since those are adapted to drier environments. But our main takeaway here was that species identity was the most influential factor in determining drought response rather than those broad community classifications. Overall, cover usually did not decline until 2 to 3 weeks into drought conditions, and most of the species were able to recover from up to 3 weeks of drought. Some species, like the inland saltgrass, performed particularly well and actually had its most cover after the most severe drought. And alkali bulrush also showed a lot of capability for recovering from drought, which indicates these species might be good choices for revegetation in a site that is prone to drought or when water availability throughout a season is uncertain. But we're going to move on to the field experiment, where the main research question was looking at whether a precision or a bed hedging seeding approach is more effective at establishing native vegetation. So 2 frameworks have been described in restoration literature as potential paths for improving restoration outcomes, especially in a changing climate. So the first, a precision restoration framework, focuses on tailoring your methods to specific site conditions or specific barriers that are present at a site. So in a revegetation context, this may look like choosing species for revegetation that have hydrologic tolerances that match the site conditions or the expected site conditions. The other, a bed hedging approach, also called a portfolio effect, it is assuming that environmental conditions are going to be too variable and unpredictable to effectively target your methods on such a fine scale. So the goal is to broaden your approach to try and mitigate risk. So in a revegetation context, this would look like seeding with a broader array of species that have a range of hydrologic tolerances, so that regardless of the hydrology at the site, something in that mix is likely to establish. So both of these frameworks have been described as conceptual restoration planning tools or studied in other ecosystems like rangelands or forests, but there's a lack of research on these methods in wetlands, especially precision restoration, and there's no direct comparisons of these frameworks in the revegetation context. So again, our goal was to determine if a precision approach or a bed hedging approach would result in more robust native plant communities. So each one of these boxes represents a treatment in the field experiment. The unseeded control was just to record species that are growing in the background of the site. And then our precision treatment is where, again, we're seeding with species whose hydrologic preferences are matching the site conditions. So in this treatment, we just seeded with 5 species whose hydrologic niches matched site conditions at the time of seeding. And this was just seeded once when water levels were optimal for germination. The next 3 boxes represent the bed hedging treatments. The first is the diverse mix treatment, where we just seeded once, again, when water conditions were optimal for germination, but we used a much higher diversity mix. This was a 22 species mix versus 5. And this mix is including species with a much broader range of hydrologic tolerances. So regardless of site conditions, we're thinking something in that mix is likely to establish. The next 2 use the same diverse seed mix as the diverse mix approach, but add another element of risk mitigation through varying the timing of germination or seeding. So the delayed germination treatment only had half of each species treated to break dormancy. So this is giving those untreated seeds the opportunity to either germinate later in the season, or if they remain dormant throughout the winter, germinate in the following year, when there's potential for conditions to be better for germination or for seedling establishment. And then in the multi-year sowing treatment, all of the seeds were treated to break dormancy, but only half of the total quantity of each species was seeded in 2024, and then the other half was seeded in 2025. So again, this is just introducing another window for germination. So in case conditions ended up not being good for seedling establishment in the first year, you have another opportunity in the second year. And we predicted that in saturated or inundated soil conditions, the precision treatment would produce the most native cover. And in an environment with less water, or if hydrology fluctuates throughout the season, we predicted that one of the three bed hedging approaches would produce the most native cover. However, of the three bed hedging approaches, we predicted that the multi-year sowing treatment would be the most effective in establishing native cover due to having those two targeted windows for germination. These are the 22 species that were used in the bed hedging treatments in this experiment. So it spans 4 different plant community types, including the 3 that I talked about earlier, emergent, meadow, and playa species. And then we also included 1 upland species here. So this is not specific to wetlands, but it is commonly found in Great Salt Lake wetlands. These 5 species highlighted in red were used in the precision treatment. So these were chosen because we also know that they're widely available for restoration practitioners in this area and have been high performers in restorations around Great Salt Lake. And these are classified as emergent or meadow species, which like to either have standing water or saturated soil, and their hydrologic tolerances aligned with our site conditions when we seeded. So we hand seeded these 2 seed mixes into 4x8 meter plots in a randomized complete block design with 6 blocks. And I sampled from 2 1x1 meter subsampling plots within each of those 4x8 meter plots. And we also installed groundwater wells at the block level to monitor the groundwater depth through both growing seasons. The experiment was fully replicated at 2 sites, Howard Slough and Farmington Bay waterfowl management areas. And these are both on the southeast arm of Great Salt Lake. These wetlands are impounded with water control structures that allow wetland managers to control the direction and the volume of water flows throughout the site. And our plot areas were in emergent wetland habitats where the soil was fully saturated at the time of seeding. I collected visual percent cover data biweekly from the beginning of the 2024 growing season and also through the 2025 growing season. And then we also monitored the groundwater depth biweekly. And we also wanted to compare the logistical requirements of all of these approaches, since we know that cost and labor availability is a big barrier for managers when they're implementing revegetation projects. So we tracked the cost of both of these seed mixes using various invoices from some seed vendors that we work with. And then we also tracked all person hours associated with implementing these treatments to determine which one is the most time intensive. So before we talk about the plant results, we can look at the hydrologic conditions because this is overwhelmingly what dictated our results between both sites. So these plots are showing the groundwater depth at each site with the monitoring date on the x-axis and then depth on the y-axis. And that black horizontal line is representing the soil surface. So at Farmington Bay, when we seeded in 2024, we did have saturated soil, but there was a really rapid drawdown after seeding. And about 2 weeks after seeding, conditions got very dry and remained very dry for the rest of the summer, except for that little spike that we had at the very end of the growing season. And in 2025, conditions were even drier, and our groundwater wells were actually empty for most of the growing season. So the groundwater depth was over 1.25 meters below the soil surface. However, at our other site, Howard Slough, we had saturated or inundated soil through the entire experiment. So we had some moderate fluctuations between monitoring dates, but overall the water levels were stable, and we had a lot of moisture for suitable plant growth. So at Farmington Bay, where we had the really low soil moisture conditions, unsurprisingly, there was very low plant growth across all treatments. These plots are showing the cover over time in 2024 on the top and then 2025 on the bottom in 3 different categories: the invasive growth in orange, seeded growth in green, and unseeded growth or unseeded native growth in blue. The x-axis is our monitoring date and the y-axis is our mean percent cover. And as you can see in 2024 in that top panel, we had almost no plant cover in all categories. Even our invasive cover was really low. In 2025, we had a small spike in seeded cover in June, but after our site conditions got really dry, the seeded cover really dropped off. And these are the raw data plots, but ultimately with our models, there was no significant effect of seeding treatment in the model results. Despite that, we did have a few native species that held on in, albeit very small numbers. But I did find it significant that there were some species that did persist through those super dry conditions. So we had some native cover from the golden dock in 2024. And in 2025, we had cover from the inland saltgrass and perch seepweed, which those are both known to be drought-tolerant species. And the inland saltgrass was our best performer in the greenhouse experiments as well. Our results at Howard Slough looked very different. And as I mentioned, we had consistently saturated soil, which was really good conditions for plant establishment. Over both years, we had steady growth of the seeded species throughout the growing season. And in 2024, the seeded cover in the precision treatment surpassed invasive cover by the end of the growing season. And in 2025, all of the seeding treatments, except for delayed germination, ended up surpassing invasive cover with their seeded cover, though the delayed germination treatment was very close. So these are our mixed effect model results of the end of season cover at Howard Slough. So this is testing the effect of seeding treatment on cover and the final monitoring date of 2024 and 2025. So our model predicted mean cover is on the y-axis, treatment is on the x-axis. Points in red are the 2024 data, and points in blue are the 2025 data. And these total seeded cover panels that are highlighted here are only modeling the species that were in our seed mix. So the unseeded control is not included in this model, as we could not accurately model that treatment with the rest of the seeded treatments because the cover values were so different. So in 2024, the precision treatment produced the highest seeded cover, but in 2025, there were no significant differences between seeding treatments in the seeded cover. So this tells us in the second year, those temporally delayed treatments, which was the delayed germination and multi-year sowing, caught up in cover to treatments that were all germinating at the same time. But in the fourth year, the precision approach, in the first year, the precision approach led to rapid plant establishment that we didn't see from the vet hedging treatments. These middle panels are the total native cover. So this is including native species that we did not seed so that we can compare it to the unseeded control. So those were just plants growing in the background of the site already. And as you can see, the unseeded control had significantly lower native cover than all seeded treatments in both years. So we did see a benefit of seeding regardless of what the treatment was. These last two panels are invasive cover. In 2024, the precision and diverse mix treatments most effectively suppressed invasive plant growth, but in time, the time-delayed treatments the time-delayed treatments did not as effectively suppress invasive cover in year one. But in 2025, the only seeding treatment to have significantly lower invasive cover was the multi-year sowing treatment. So even in this environment with suitable water availability, even though this treatment was designed to mitigate risk in a hydrologically extreme scenario, in this environment, repeated seeding still gave us a cumulative benefit, even though in total it was the exact same amount of seed going out onto the landscape in this treatment than the other ones, but seeding in two separate events gave us a benefit of more native cover. So even though the delayed germination treatment also had additional time windows for germination, it may not have been as effective as the multi-year sowing because seeds that were dormant might have been exposed to predation or unfavorable weather. So we did not see the same cumulative benefits with that treatment. So this plot is showing the breakdown of the mean cumulative cover of all species in the final week of the experiment. With 2024 on the top and 2025 on the bottom. And we saw our native species composition change between years, with most of our seeded treatments gaining species diversity in the second year. So in 2024, these 4 species were the main drivers of our native plant cover. So the beggar's tick and the golden dock which are both annual forbs that did well in our greenhouse experiments, as well as the inland saltgrass and hardstem bulrush. So in 2025, those species were still present in our plots, especially the beggar's tick in large amounts, but we also had several species come up from our seed mix that did not appear in 2024. So this is the common spike rush, the scratch grass, the seaside arrowgrass, and then we also had a greater quantity of this chairmaker's bulrush in the second year versus in the first year. So these are the results showing the cost of the seed mixes as well as the relative time investment that was required by each approach. So the blue bars are a relative time investment and the green bar is the cost of the seed mix. The precision seed mix ended up being about $17 per pound cheaper than the diverse mix, though again, there was a vastly different level of species diversity between these mixes. It was 5 versus 22 species. So this is not a huge difference for one bag of seed, but when managers are working on large scales that require thousands of pounds of seed, that difference could become prohibitive if you're trying to incorporate species diversity. So these time estimates are based on our experimental process for seeding. So this includes breaking dormancy, weighing seeds, and then the actual installation of the experiment. So actual tasks are going to vary for restoration practitioners, which is why we're using the relative time. So the precision and diverse mix treatments took about the same amount of time to implement, so that is our baseline. But the delayed germination treatment took about 1.5 times the amount of time, and that extra time is accounting for weighing stratified or dormancy broken seed versus non-stratified seed. And again, in an actual restoration scenario, those time investments are going to vary. Multi-year sowing gave us the greatest ecological benefits, but unsurprisingly, this method also required by far the greatest amount of time to implement at 3 times our baseline due to needing an entire separate seeding event in the following year. So our main takeaways from the field experiment results are that sufficient water is critical in establishing native vegetation. And even if you are using drought-tolerant species, if a site is super dry, like we saw at Farmington Bay, even those may not be able to establish. The precision treatment gave us the highest seeded cover in year 1 at Howard Slough, but by the second year, multi-year sowing gave us the greatest ecological outcomes in suppressing invasive growth. So based on these results, we're going to go through some management recommendations. So based on my greenhouse experiment results, I'd recommend that if 3 or more weeks of dry soil conditions should be avoided after seeding to support plant establishment. So as I mentioned, these wetlands have extensive water control structures. So if there is the potential for moving water around to support seedlings after or when you want them to germinate, any more than 3 weeks of dry soil conditions is going to significantly affect their growth. And if water control to that degree is not possible, seeding with species that are able to tolerate or recover from drought may improve revegetation outcomes. So this would be species like the inland saltgrass that had its highest cover after 4-week drought, or the alkali bulrush that was also able to recover to similar cover values across treatments. And then thinking about the field experiment results, even seed mixes designed to buffer against hydrologic stress can still fail in extreme moisture limitation. So this would warrant avoiding seeding of sites that have the potential to dry down to extreme levels like we saw at Farmington Bay. And at sites where soil is saturated, or you would just expect consistent hydrologic conditions throughout a growing season, targeting species to those site conditions, like we saw with the precision treatment, could aid in early native plant establishment. However, repeated sowing is going to give cumulative benefits due to allowing for different windows for germination. However, this was also the most labor intensive. If those resources are available to spread seeding over multiple years, this could lead to greater outcomes, but again, resources might not be available. Okay, before we get to questions, I have my thank yous. So I would like to thank Karin, my advisor, for her support and guidance the last 2.5 years. And then I'd also like to thank my committee members, Tom Monaco and Tim Walsworth, for their really helpful feedback. Thank you to Keith Hambrecht, Rich Hansen, and David England of Utah DWR and Forestry, Fire, and State Lands for coordinating with us on our field experiments. And also thank you to Alec Hay and the rest of the greenhouse staff for their help in getting my greenhouse experiments up and running. And then thank you to Susan Durham, Calvin Bladen, and Alice Carter for their statistical input into these experiments. And then I'd also like to thank the Wetland Ecology and Restoration Lab for all of our wonderful field times together. Okay, and now I will take questions. Oh, yes.
Ask you some version of this question before. But it's interesting that your ecological groupings don't respond as strongly as individual species.
Mm-hmm.
There's an awful lot of grouping going on out there in science world.
Mm-hmm.
What's your take of that? Do you think this is a general phenomenon and not restricted to wetland plants? Do you think we've oversold the idea that we can classify species into guilds or whatever we're using and expect to see more interpretable responses by doing that? It's a big issue. I'm just curious as to what your take is and whether we ought to continue to pursue using that approach.
Mm-hmm. I think it could be, at least with the plant groupings, it can be useful to have those classifications just because there are different environments are so different and you are going to find like different communities. But when you're thinking about implementing things like in a restoration scenario or really specific treatments, I do think there are a lot of differences between species, and then just reducing it down to the group could lead to outcomes that you're not expecting or that are not ideal, just because there is a lot more nuance in how individual species, plants or animals, are going to respond to things. So I think there's value in the groupings if you're talking about like general trends, so like the general environment that these species grow in. But if you need like specific information or you're trying to do a specific like restoration treatment, then going down to finer levels like functional groups or species would be more beneficial.
The crappiness of our current groups is associated with a poor understanding of the full niches of species, and that we're just focusing on a part of each species niche, or is it something different?
I think at least like here specifically, like these groupings are based on the types of environments that the plants grow in, but you could also group by like functional traits or other things. So sorry, could you repeat your question?
I think I forgot.
Okay. Yeah, ultimately, I think like there's some value in the groupings, but it depends on the way that like what you're looking at.
Do we do a crappy job at actually measuring the overall niche, all the important dimensions? Or is it mostly because we know a few dimensions or maybe only one dimension, so we group by that and ignore everything else that's important?
I don't know much about ecology groupings outside of wetland plants, so I feel like I can't speak to in general if ecologists are doing a bad job. But I think here, like in this sort of scenario, again, it depends on what you want or like what you need. Yeah. Okay. Yes, Ed.
I don't know how to phrase this question proper, but in your second experiment, when you look at the different plots and you looked at the slough, that was the one situation in which you were getting native plants that you were trying to restore growing more than invasives. Based on that, and I think you sort of almost skirted around this a little bit, but when you've got these impounded wetlands where you actually have the ability to control some of the water that's going in, think about how something like the Berwick Migratory Bird Refuge is managed now. What recommendations would you then make in terms of how to spread or not water around the landscape?
Yeah, so these wetlands are primarily managed for waterfowl. So they're trying to create good habitat and like food for waterfowl each year. So a lot of times even if we are doing revegetation, the goals for the water management, sometimes there's a disconnect between what we need to do with the water and then what the areas that we're seeding. So there's a lot of limitations that these managers work with. But if water is available, like it was at our Howard Slough site, like we had a lot of excessive water coming in, I would recommend making sure that the site is saturated during seeding and then for several weeks after for early establishment. But if water needs to be moved around towards the end of the season, waiting until those plants are in a more robust state might lead to greater outcomes because they might be able to push through the rest of the season, even if it does get dry. And then if they're perennials, like the rootstock will survive over the summer. At our Farmington Bay site, that's kind of what our situation is with the water there. We're near an outflow where water comes in at the beginning of the season, but they divert that water for waterfowl towards the end of the season. So if there is flexibility, I think keeping a steady stream of water at the beginning for early establishment is really important, but if the management goals are conflicting, moving it later in the season might still give the opportunity for those plants to germinate.